Hey, and welcome back, everyone. So let's go ahead and finish up the last chapter in our cardiovascular unit. So if you recall, chapter 18 was all about the characteristics and composition of the blood, including the different types of chemicals that you find in the blood plasma, different types of formed elements and the life cycle that they go through, the transport of oxygen by hemoglobin. Chapter 19 was about the function of the heart itself in pressurizing blood in order to pump it throughout the circulation. We talked about the effects of the autonomic nervous system and its ability to regulate the cardiac output. So the last part of this unit, chapter 20 here, is specifically going to focus on the blood vessels, all the arteries and veins, and all the other types of vasculature that are going to function in carrying blood to different places throughout the body and in facilitating the delivery of nutrients and oxygen and the pickup of different waste products. So let's hop right into this. The first thing to understand about this chapter, do not forget that your textbook is a combined physiology and anatomy textbook. So naturally, this chapter is going to contain a boatload of anatomical information. So to help you out here, basically, we're only going to cover section 20.1, 20.2, and 20.3. Everything after that covers very gross anatomical concepts in terms of all the different branches of the aorta, all the venous drainages that lead into the vena cava. So if you haven't already had an anatomy class, you will get that in anatomy. And if you have had anatomy, you've already had that. So regardless of your station in whether you've had anatomy or not, we are not going to cover that. We are going to just focus on core concepts here as it pertains to physiology. So back in chapter 19, at the very beginning, we covered the difference and distinctions between the systemic circuit and the pulmonary circuit. Just as a reminder, blood that is in the pulmonary circuit is on its way from the right side of the heart to pick up oxygen in the pulmonary capillaries around the lungs, and then it's on its way back to the left side of the heart. Blood that is in the systemic circuit is on its way from the left side of the heart out to all of the tissues and organs that need oxygen and need nutrients, and then on its way back to the right side of the heart. So we also covered some definitions regarding blood vessels back in chapter 19, just some very preliminary definitions. So we said that arteries are always going to carry blood away from the heart. So we said that usually the case is that this blood will be oxygen rich. Usually there is one exception. And veins are going to return blood back to the heart. And again, the general rule is that this blood will typically be deoxygenated. So one thing, and then the capillaries that exist in between the arteries and the veins are going to be the site of gas exchange. And the point that I made back in chapter 19 is you can tell that the capillaries must be where the gas exchange occurs because based on the way pictures like this are colorized, with a red color indicating oxygen-rich blood and the blue color indicating oxygen-poor blood, the fact that we get this kind of purple color as we transition from red on the arterial side and blue on the venous side, as we see the red color disappear to blue color, this tells us that these capillaries must be where the oxygen pickup and delivery occurs. So I mentioned the two exceptions to the oxygenation rules from above. We covered this in chapter 19, so if you don't quite remember this, I suggest you go back to chapter 19 and review that. So let's take a quick peek at the structure of different types of blood vessels. So any given blood vessel can have up to 3 different histological layers. We sometimes call these layers tunics. So these 3 different tunics are each going to serve a slightly different purpose in either maintaining the blood vessel, permitting blood flow, or something else altogether. So the first type of blood vessel tunic is called the tunica externa. So that externa part of that basically tells you that this is the outermost layer of the blood vessel, externa, external, right? So basically, this is just a layer of fibrous connective tissue that kind of is the outermost coating of the blood vessel itself. More or less what this is here to do is to connect the blood vessel to the underlying extracellular fluid and extracellular matrix to make sure that we anchor it down in place as part of the tissue or organ that it is supplying blood to. So basically, in most cases, we don't want major arteries and veins flopping all over the place. We kind of think of them as kind of like slippery noodles, but because of this tunica externa, they are rigid's not the right word, but they're pretty stringently laid down in place, and they're not really going to go anywhere, which is a good thing for us. The middlemost layer, called the tunica media, media meaning middle, this is a layer of vascular smooth muscle, and this is something that, even though we have not actually formally named it until now, this is something that we are already very familiar with based on our discussions of the autonomic nervous system, particularly the sympathetic division's ability to vasoconstrict and vasodilate blood vessels as a means of regulating blood flow into and out of specific places. So we talked about the fight or flight response. We vasodilate the smooth muscle in the tunica media of blood vessels that feed skeletal muscle, while we vasoconstrict the smooth muscle in the tunica media of blood vessels that feed the intestines and feed the kidneys, right? So we've talked about this sort of thing before, so now we're just giving that layer of smooth muscle a name. And then the tunica intima, or sometimes also called the tunica interna, like internal, this tells you this is the innermost layer of the blood vessel. This is just a layer of vascular endothelial cells of the squamous variety. So basically, this is just the innermost layer of the blood vessel, and basically, if you kind of think of a blood vessel as a hollow tube, this is what the tube actually is. So this is the actual layer that interacts with the blood on the luminal side of the blood vessel. So this is just this layer of vascular endothelial cells along with their basement membrane and a layer of elastic tissue to allow for some compliancy when a great volume of blood enters into that particular blood vessel. So even though there are 3 different possible tunics for every blood vessel to have, not every type of blood vessel is going to have all 3 of them. But every type of blood vessel that we are going to talk about, at minimum, is going to have the tunica intima. So every type of blood vessel has to consist, at minimum, of this layer of vascular endothelia. You don't necessarily have to have the other 2 in order to constitute a functional blood vessel. So our 5 different types of blood vessels that we have mentioned before are arteries, arterioles, capillaries, venules, and veins. So what we're looking at here is the breakdown on how much of each tunic does each particular type of blood vessel have. Well, let's start with the types of blood vessels that have all 3 of the tunics. That is going to be arteries and veins, the 2 largest types of blood vessels. So you can see here in this breakdown that arteries, of course, have the tunica intima, this layer of vascular endothelia. In fact, you can see all of these blood vessels have that, just like we said on the previous slide. And in addition, it also has this layer of elastic tissue, which sometimes, but not always, goes along with the tunica intima. Arteries have the vascular smooth muscle, like we said. So do arterioles and veins. And then arteries, venules, and veins also have the tunica externa, that layer of fibrous connective tissue that connects us to the extracellular matrix. So let's take a look at a couple of omissions here in terms of the tunics. So most notably, we said that every type of blood vessel is going to have the tunica interna, right? So every type of blood vessel has this layer of vascular endothelia that you can see here on the innermost part and luminal side of the blood vessel, where the blood is going to be contained. So if we're talking about smooth muscle, you'll notice here that the only types of blood vessels that have this tunica media, the smooth muscle layer, are arteries, arterioles, and veins. Capillaries and venules do not have any smooth muscle. What that tells us, by definition, is that we cannot vasoconstrict and vasodilate capillaries and venules because they do not have vascular smooth muscle. So if you're thinking of trying to regulate, restrict, or promote blood flow at the capillary or venule level, you better think again, because you cannot do vasoconstriction or vasodilation if that smooth muscle does not exist. And what we are going to see eventually is that most of the functional vasoconstriction and vasodilation that we've talked about before is actually going to happen at the arterial level, and to a much lesser extent at the artery level. And we'll discuss a little bit later why vasoconstriction and vasodilation probably isn't going to be the most effective thing if you do it at the vein level. Another noteworthy feature you'll notice here is that capillaries literally are just a layer of tunica intima. All capillaries are is just a hollow tube that is made up of vascular endothelial cells. Capillaries do not have smooth muscle. They do not have the tunica externa to anchor it down into a tissue. So capillaries are as simple a type of blood vessel as you are going to find. It is literally just a hollow tube, a very narrow hollow tube that has a mean diameter of just 8 microns as compared to veins and arteries, which are almost 1,000 times wider than a capillary is. So capillaries are just very, very narrow hollow tubes that are made up of vascular endothelial cells. In fact, as a little bit of trivia here, capillaries are so narrow in terms of their diameter that they are only wide enough for a single red blood cell to fit through a capillary at a time. So as blood circulates and makes its way through capillary beds, red blood cells are literally going in single file line through the capillary openings. So if you look at this picture down here in the bottom left, you're going to notice a direct comparison of an infiltrating artery into whatever section of tissue this is. I can't really tell what organ or what tissue this is taken from. So you'll notice an infiltrating artery bringing oxygen-rich blood in, and then you'll notice an exfiltrating vein coming back out, bringing the oxygen-depleted blood on the other side of whatever capillary bed is being serviced to this tissue here. So obviously the first thing that you're noticing here is the fact that the artery kind of looks like a perfect cross-sectional circle here, and then the vein almost looks like it's totally collapsed upon itself. So you might be wondering, are we looking at some kind of disease here? Is there something wrong with this person's veins? No, this is actually exactly what we expect. And it gets down to something that we talked about in chapter 19 as it pertains to pressure gradients. You'll remember that blood circulates throughout our bodies from high pressure situations to low pressure situations. Also, blood moves from arteries to capillaries to veins. Naturally, arteries are going to be at a much higher pressure than veins. So the fact that you see an intact artery here is attributable to the fact that it is carrying highly pressurized blood, and that pressure being placed on the blood is able to make sure that the artery does not collapse upon itself, whereas the vein here is carrying very low pressure blood. So that vein, because it's carrying very low pressure blood, has a tendency to kind of push down and collapse on top of the blood that is being carried through there. But also, in addition, veins tend to be a lot more elastic than arteries, so a vein isn't likely to look like this all the time. The vein actually has the ability to greatly expand itself away from this collapsed state to accommodate a much greater volume of blood coming through. So veins are a little bit more dynamic than the arteries are. So just to kind of briefly go through some of the things that I've already talked about, arteries and arterioles are going to contain all three of the tunics that you can see here. So you can see several different types of arteries here, and then the arterial right there. I mentioned that most of our vasoconstriction and vasodilation will happen through the arterioles rather than through the arteries, although it does happen a little bit in the arteries. So I also mentioned that veins do have a tunica media, so they do have that vascular smooth muscle. But one thing that I'd like you to think about in advance of our next meeting is what do you know about veins that would tell you why vasoconstriction and vasodilation might not be so effective there? So what you're going to want to think about for that question, and it is kind of a head scratcher, what you're going to want to think about there is what is the purpose of vasoconstriction and vasodilation to begin with? What are we trying to accomplish, say, in the fight or flight response in terms of getting extra blood flow to skeletal muscle at the expense of taking blood flow away from the GI tract and the kidneys? So think about whether we would be able to accomplish that if we did our vasoconstriction and vasodilation at the level of the veins. So make sure you think about that. So this is a picture that I really, really love. So this is actually not from your textbook. It's actually from the D. Unglob Silverthorne textbook that I used to teach out of, but I love this picture, so I thought I'd go ahead and include it for you. So in this picture, you can see kind of a breakdown of a very simplistic and kind of broken down breakdown of the circulation that we've talked about before. So just to kind of very briefly go through this, you've got the right side of the heart, pulmonary artery into the pulmonary capillaries where we get gas exchange and we pick up oxygen, through the pulmonary veins to the left side of the heart, out through the aorta into the systemic circuit, and then the aorta will branch off into our major arteries, both ascending and descending, which will then service down into different arterioles, capillary beds, venules, veins, and then back to the vena cava. But what really makes this picture great is what is being illustrated here with these different capillary beds. So each one of these 4 capillary beds is meant to basically be the blood supply for various different organs. So you could maybe think of one being the brain, one being the heart itself, and you could consider these to be your pulmonary arteries. One could be the kidneys, and one could be the liver. So you can use your imagination there. So I had mentioned that vasoconstriction and vasodilation very often happens at the level of arterioles. So what makes this picture so great is the fact that the ability that we have to vasoconstrict and vasodilate arterioles is indicated by these little adjustable screws that you see right here. So you can imagine that if you want to cut off blood flow, well, you simply need to tighten one of those screws to narrow the diameter of that arteriole and not permit any blood to flow through. So it's kind of a thought exercise here. If you say on the very left-hand side have your brain here, and on the 3 capillary beds to the right, you have things like the kidneys, the liver, and the GI tract. Well, if you, for some reason, needed to greatly promote blood flow to the brain, think about how much blood would be flowing in to the brain if you were to vasoconstrict the 3 arterioles that lead into those at least for the time being, non-essential tissues. So another way to think about this is that if there is no vasoconstriction and these arterioles are just wide open, then if, for the sake of argument, just for this thought exercise, if the aorta up here is carrying what we'll call 100% of our blood flow, right? So 100% of blood flowing into the systemic circuit. If these are the only 4 tissue capillary beds that are being serviced by the aorta, then if all of our arterioles are wide open, then each of those capillary beds is going to receive 25% of the overall blood flow, right? With me so far? Okay, so what would then happen if we vasoconstricted one of those? So let's say this one on the far right here. So let's say we vasoconstrict through a sympathetic signal. We vasoconstrict that particular blood vessel and completely shut it off. So no arterial blood can flow through that particular capillary bed. Well, these 3 capillary beds on the left here are now no longer receiving 25% blood flow. They have now gone up to 33 1/3. Okay, what if we then closed off this arteriole here with vasoconstriction? Well, now the two on the left, instead of 25 or 33 and a third, they're now getting 50% blood flow. So again, not that this is a new concept for us, but this picture is just so good at illustrating what you can accomplish with vasoconstriction and vasodilation. I feel like it would be a disservice if I did not provide it to you. So I hope you kind of appreciate just how good and how helpful this picture is. Okay, so capillaries, I mentioned, are more or less just a single hollow layer of vascular endothelia with a basement membrane to help us attach it down to the extracellular matrix. So capillaries, as we mentioned before, are the site of blood delivery to tissues, which is called perfusion, and they lie in between the arterioles and the venules on the artery and vein side of the capillary bed. So capillaries, like we said, only consist of the tunica intima, meaning the vascular endothelia, along with the basement membrane that comes along with the, with the territory. So capillaries come in several different forms. We've actually already covered the first two that you see here. Continuous capillaries exist mostly in the blood-brain barrier, meaning the capillaries that infiltrate the spinal cord and the nervous tissue of the brain. So because the vascular endothelia are sealed together with tight junctions and also with astrocytes helping out, that again is what forms the functional blood-brain barrier, meaning that anything that is going to perfuse between the blood plasma and the nervous tissue has to be something that either is freely permeable to plasma membranes or has some type of carrier to let it across. Most of the blood capillaries in our body are of the fenestrated variety, meaning that there are little gaps in between the edges of the vascular endothelia because there are no tight junctions holding them together, so these are fairly leaky. And then you don't really need to worry too much about the sinusoid capillaries. We have not talked about them very much, and we likely will not talk about them. So here's something kind of interesting. So this is a picture of an arteriole leading into a capillary, and then coming out on the other side, we have a venule. So capillaries, as I mentioned, are extremely narrow. They have such a small diameter that that red blood cells can often only go through in single file line. So there's nothing really new here except for one thing. Capillaries are usually often the target of microhemorrhages, meaning that it's not really that unusual for some type of injury to occur that ruptures different capillary beds. So it sounds like a big deal, but it's really not. So we can reestablish capillaries fairly easily. But you don't want to be totally dependent on capillaries for continuous circulation of blood, which is why oftentimes you will see these kind of detours here that directly connect the arterioles to the venules, and these are called anastomoses. So basically just an extra thoroughfare of blood that skips over the capillary bed and ensures a continuous circulation of blood, meaning that if you do somehow destroy the capillary bed, it's not going to completely stop the flow of blood throughout that particular part of the body. Okay, and then venules and veins, comparatively speaking, are relatively thin, really pretty wide in terms of their diameter, and we mentioned that because they are carrying very low pressure blood, they tend to kind of collapse in on themselves. So they're quite elastic, very thin, again, indicating their propensity to collapse in on themselves. And then we said veins have all three layers, and then the venules are only going to have the tunica intima and the tunica externa. So the tunica media is negligible there, to say the least. So something interesting that comes along with the territory of venules and veins carrying very low pressure blood is that with arteries, we can get arteries to pump blood from the bottom of our body up, meaning that if you're talking about from the heart, which is in the center of your thoracic cavity, blood can be pumped up to your brain against the force of gravity just because that blood is so highly pressurized. You run into a problem with the same concept when you're talking about the veins. Say you're talking about veins in your legs. So what you understand these veins need to do is they need to get that very low pressurized blood up your legs, up your chest, through the inferior vena cava and back to the right atrium. But that's kind of a hard sell because that blood is very low pressure and it's having to fight against the downward force of gravity. So that's something that's going to be kind of hard to accomplish. So to help us with that, veins, not the case with arteries, but veins come equipped with backflow valves. So basically these valves allow blood to kind of inch forward every now and then, and these valves will open and snap shut to prevent blood from coming back down with the force of gravity. And because we have very low pressure blood contained within the veins, the blood flow is going to be relatively slow here, which ultimately is going to mean for us that the majority of the total 5 to 6 liters of blood in our body, the majority of that at any given time is going to be in the veins rather than the arteries. So here you can see a table that just kind of does a quick breakdown between the arteries and the veins, comparing and contrasting them. We've covered most of this. We've covered the conduction of blood in terms of direction. We've covered the whole rounded versus collapsed concept, high pressure versus low pressure, thick versus thin. So we've covered all this. You may just want to use that as a study guide moving forward. Okay, so our next concept is something that if the coronavirus thing wasn't a thing, we would be doing an extensive lab on blood pressure measurements. But because of that, we had to kind of scrap that and move things around. But I still want to make sure that I'm giving you all the theory behind blood pressure and how blood pressure is measured. So let's talk about blood pressure here for just a few minutes. So obviously the blood pressure lab reference here, that's not happening for reasons I just mentioned. So just pretend that kind of preamble there does not exist. So when you are measuring blood pressure, the first thing that I should make sure is clear when someone says, oh, my blood pressure this, my blood pressure that, my doctor measured my blood pressure, I'm on blood pressure medication. When people colloquially mention their blood pressure, what they are specifically referencing, whether they're aware of it or not, is their arterial pressure. Anytime you talk about, oh, my blood pressure is 120 over 80, my blood pressure is a little high, it's 135 over 85, anytime you're talking about your blood pressure like that, you are specifically talking about your arterial pressure, not your venous pressure, not your pressure anywhere else in your body, just in your arteries. So if you are talking about your arterial pressure, you are, I'm sure, are aware the pressure is going to toggle back and forth between a higher pressure number and a lower pressure number. The higher number is called the systolic blood pressure, and the lower number is called the diastolic blood pressure. And if those two terms seem familiar based on what we talked about in Chapter 19, that's because they are directly referencing systole, the period in the cardiac cycle in which the ventricles are contracting and ejecting a stroke volume, about 80 milliliters of blood out into the systemic circuit, and diastole, in which the ventricles are relaxed and not currently in the process of pumping out blood. So again, when you're talking about your blood pressure, we specifically mean your arterial pressure. We know we do not mean the venous pressure when we just say blood pressure, because veins are going to, as we are aware now, carry very low pressure blood. Your venous pressure is usually going to be somewhere in the neighborhood of 2 millimeters mercury, so about 50 times less than your average arterial pressure. So you definitely do not mean the pressure in your veins when you are colloquially referencing your blood pressure. So the pulse pressure is the pressure felt on an artery when that particular artery expands following ventricular systole. So this is going to help us understand why the systolic pressure is always higher than the diastolic pressure. So the systolic pressure in the arteries is that higher pressure number, and it is the result of the ventricles, which have just gone through systole, pumping out an extra roughly 80 milliliters of blood. So if you think about an artery that is about to expand to accommodate that extra volume of blood that's coming from the left ventricle. That extra pressure is what takes you from the diastolic pressure up to the systolic pressure. So the pulse pressure is just the additional pressure that your arteries experience when they expand to accommodate that stroke volume of blood. So naturally, the pulse pressure is just the difference between the systolic and the diastolic pressure. So for example, if your systolic pressure is 120 millimeters mercury and your diastolic pressure is 80 millimeters mercury, generally accepted normal blood pressure readings or average blood pressure readings, the pulse pressure, which is very easy to calculate here, would be 40 millimeters mercury, just the difference between those two numbers. So what that essentially means is that if you manually take your pulse, whether you use the radial artery in your wrist or your carotid artery in your neck, what you are feeling every time you feel your pulse is you are feeling approximately an extra 40 millimeters mercury worth of pressure as that stroke volume of blood courses through that artery and across your finger. You are feeling that blood being delivered from the left ventricle through the aorta and into that particular artery. And like I said, the pulse pressure, it's just that increase in arterial pressure that results from the delivery of that stroke volume being distributed throughout that particular artery. So it therefore represents the expansion of arteries from ventricular systole. So the mean arterial pressure, or MAP, is going to be just the average arterial pressure during the cardiac cycle. So what you're probably thinking here is that, okay, if my diastolic pressure is 80 and my systolic pressure is 120, then the mean arterial pressure, and mean obviously just means average, the mean arterial pressure is obviously just going to be the average of the two numbers, right? So your mean arterial pressure is 100, right? So 120 plus 80, that's 200, divide it by 2, that gives you 100. Well, that's not quite how mean arterial pressure works. So just because you average the two numbers does not mean that on average that's what your arterial pressure is most of the time. There's something that we have to take into account. The fact that your ventricles spend more time in diastole than they do in systole. They spend more time resting than they do contracting. So we need to account, therefore, for the fact that your arterial pressure spends more time at the diastolic number than it does at the systolic number. So the way that we can calculate this, it's a little bit more of a hassle to calculate rather than just adding the two numbers together and dividing by 2. You take the diastolic pressure plus 1/3 of the pulse pressure. So if we use the 120 over 80 figure that we used in the previous slide, 80 millimeters mercury plus 40 over 3 would give us a mean arterial pressure of approximately 93.33 millimeters mercury. So if your diastolic pressure is 80, your systolic pressure is 120, a perfect average of those two numbers, like I said, would be 100, so the fact that your mean arterial pressure is closer to 80 rather than 120 is reflective of, again, the fact that your heart spends more time relaxing than it does contracting. And being aware of the mean arterial pressure actually ends up being highly significant because we understand that a high arterial pressure is what is necessary to drive blood flow through the arterioles and through the capillaries and to get blood into the veins so that we can eventually start this whole process over again. So we've talked about ischemia in the previous chapter, so it's important to at least be aware that not having a sufficiently high mean arterial pressure is going to result in ischemia, the lack of blood flow to affected tissues. So you can see right away why low blood pressure is going to be kind of a big deal there. So in this picture, you can see the way that blood pressure in different areas of the body can tend to fluctuate a little bit. So if you start on the left side of this diagram here in the left ventricle, you're seeing exactly what we talked about in the cardiac cycle in chapter 19. You can see down here at the very bottom where we're at the very low pressure, when the pressure rises, when the pressure rises, you are going through isovolumetric contraction to raise the pressure up so that we can eject the stroke volume. And then we go through isovolumetric relaxation to fill back up from the atria. Isovolumetric contraction again, isovolumetric relaxation. Once we get out into the arteries, though, you can start to see we're going to start toggling back and forth between the systolic pressure, which here is indicated by 120, and the diastolic, which is 80. So we just keep toggling back and forth. Systolic pressure when the ventricles are contracting, diastolic when they're relaxing. Systolic when they're contracting, diastolic when they're relaxing. But with the mean arterial pressure that you can see here by this pink line, as we start to drive that blood through the arterioles and into the capillaries, things stop fluctuating so much, and then we kind of hit a more linear region of blood pressure decline. Understanding, of course, that blood naturally circulates from high to low pressure, and we reach the absolute minimum pressure at the superior and inferior vena cava just as we're draining into the right atrium. So let's talk for a few minutes about the practical aspects of measuring blood pressure. So I think this is something that is essential to understand, especially since we now live in an era of technology in which blood pressure measurements are more done so by machines rather than actual people. You can actually just strap a cuff on someone's arm, press a button, and the machine is going to do all the work and then just spit the 2 blood pressure readings out at you. So I place a high amount of value on being able to do it the old-fashioned way, which again is what we would have done if the whole coronavirus thing hadn't happened. So hopefully you'll get some benefit out of this. So a blood pressure device that actually measures blood pressure is called a sphygmomanometer. So that's a mouthful, obviously. So what it's of is a inflatable rubber cuff that is always going to be necessary no matter what the technology is, that is placed around the patient's upper arm. We inflate it essentially to cut off the flow of blood through the brachial artery, that artery that moves down through the arm and supplies blood into the hand. So we are going to cut off the flow of blood, and the practitioner, whether it's a nurse or a doctor or someone else, is going to use a stethoscope to listen to the sound of blood moving through that brachial artery. So in practicum here, the cuff will be inflated to such a great extent, like I said, that we are going to completely halt the flow of blood through that brachial artery. So let me kind of position myself here. So if the cuff is on my left arm here, blood will be flowing from my heart out through the aorta into my shoulder, but it won't be able to move past the cuff right here. So the blood will get stopped right here and it won't be able to move into my forearm and into my hand. So before we go any further, I mentioned that the practitioner is going to be using a stethoscope to listen to the sound of blood flow. So let's talk about blood flow and the different types of sounds that it makes. So before that cuff was ever inflated, so as you are sitting in front of your computer screen right now with no cuff restricting blood flow, blood is moving through your arteries and through the rest of your body in what is called laminar fashion. So what you can see right here. So laminar blood flow means that all the different components of the blood are flowing parallel to the walls of the blood vessel, meaning everything is going to be nice and smooth, everything's moving in the same direction. So naturally, laminar blood flow, because everything is moving so smoothly, this is going to be completely silent. You cannot hear the sound of laminar blood flow. So if you have a stethoscope and you try to listen to your brachial artery right now, you're not going to hear anything. The flow of blood is so smooth, you're just not going to hear anything. But when you inflate the cuff and you start narrowing the diameter of that artery, you're going to start, because of the narrowing, you're going to start inducing what is called turbulent blood flow, meaning the same volume of blood is trying to squeeze through a much narrower space now. So as things squeeze through, things are going to kind of start shooting off in all different directions. Different parts of the blood are going to start colliding with the side of the blood vessel. These collisions are going to produce an audible sound. So if you're listening with a stethoscope and you can actually hear the blood flow, you are listening to turbulent blood flow. So let's walk ourselves through the procedure of measuring someone's blood pressure here from start to finish. So when you inflate that cuff, you're aiming to inflate the cuff higher than what the systolic pressure is predicted to be. So usually you're going to predict, so the figure here has the cuff pressure at about 140. It's usually going to be more like 160. We want to make sure we get it nice and high so that we can make sure we completely close off that brachial artery. So you can see blood trying to flow through this brachial artery, but because the pressure in the cuff is so high, blood cannot move any further beyond that. So because there is no blood flow down here where you are listening, there is no blood flow there, you can't hear any blood flow if there is no blood flow. So this is going to be completely silent at this point. Okay, so once you've gotten to that point, you are going to use the nozzle on the cuff to very slowly and gradually start releasing some air out of the cuff. And this is going to start reducing the cuff pressure. So what you are doing here is while you are releasing air from the cuff, you are using your stethoscope to continue to listen to the brachial artery. It was silent before, but as you relieve the pressure in the cuff, you are eventually going to open that brachial artery so that blood can just start squeezing through. And when that blood squeezes through, it is going to be noisy. You are going to hear the blood shooting through that opening that you are creating in the brachial artery. So as soon as you hear that first turbulent blood flow sound, stop releasing pressure in the cuff and look at your pressure gauge. Whatever the pressure was at that first sound, that is going to be the systolic blood pressure, meaning that the cuff pressure is roughly equal to the systolic blood pressure, meaning the systolic pressure was just sufficient to overcome the cuff pressure. So like I said, in most normal cases, that's going to be about 120 millimeters mercury. Okay, so that's half of our job done. So currently, when you're at the systolic pressure and you're listening to the brachial artery, you are hearing every ventricular systole. You're hearing whoosh, whoosh, whoosh. You're hearing every time the stroke volume is delivered, and you hear that turbulent blood flow every time blood shoots through the brachial artery. So now you are going to continue to listen to these turbulent blood flow sounds as you continue to reduce the pressure in the cuff. Eventually, as you reduce the pressure, you are going to get to the point where the pressure in the cuff is no longer in any way sufficient enough to restrict the flow of blood through the artery. So eventually, you are going to restore laminar blood flow. So now what you are listening to, now that you have recorded the systolic pressure, you are listening for the exact moment those turbulent blood flow sounds disappear. So once you stop hearing turbulent blood flow as you reduce the pressure in the cuff, take a look at your gauge. That pressure, when you stop hearing those sounds, is going to be equal to the diastolic pressure, that point where you have restored laminar blood flow. So as long as you are listening to the brachial artery and as long as you can hear turbulent blood flow, you know you are somewhere in between the systolic and the diastolic blood pressure. If you are above the systolic pressure, you're not going to hear anything because there is no blood flow. If you are below the diastolic pressure, you're not going to hear anything because that is laminar blood flow. So the sounds of the turbulent blood flow that you hear in between the systolic pressure and the diastolic pressure are called choric cough sounds. And there are actually several different types of choric cough sounds that we're not really going to get into, but that's what they are essentially called. And this picture that you see right here is just kind of another illustration in more kind of graphical form of how blood pressure is measured. We go from silent, no blood flow when the cuff pressure is very high to audible turbulent pressure. We're in between the first and last choric cough sounds, so we're in between the diastolic and systolic blood pressures. And then eventually we restore laminar blood flow, and we're not going to hear that either. Okay, so now that we've had a good discussion on blood pressure and blood pressure measurements, Now we can start to talk physiologically about the sorts of things that influence blood flow and blood pressure through these different types of blood vessels that we've talked about. So since we've talked about how the major pressure reading that we are concerned with is the arterial pressure can be determined by a number of different types of factors, and hopefully all of these should make some semblance of sense based on discussions that we've had in the past. The first and one of the most important factors that tells you or determines your blood pressure is your cardiac output. So the idea here is that your cardiac output, which is the volume of blood being pumped by your heart every minute, the more blood that is being pumped out, the higher the pressure is going to be. So think about if you go outside your garden and you have a garden hose, you turn on the faucet. Think about the pressure in that garden hose as you turn the water up and up and up, and you get more and more water shooting out. The garden hose is going to have to expand to accommodate all that extra water. So if your heart starts beating faster or beating harder, naturally your arterial pressure is going to come up because you have more blood flowing through those arteries. Blood volume is also a big one. So you roughly have somewhere between 5 and 6 liters of blood in your body at any given time, but of course that can change with things like dehydration, excess drinking of things like water. So if you drink a big gallon of water, that gallon of water is going directly into your blood plasma, and it's going to increase the volume of your blood and increase your blood pressure. So we'll eventually talk in the 5th unit about how your urinary system hops into action quickly to take care of that, so it's not really that big a deal, so don't feel like you have to deprive yourself of water just because you're concerned about your blood pressure. But this is another very important determinant. The volume of blood contained in your arteries is obviously a very important determinant of the pressure. Blood viscosity is not really that important or that physiologically relevant for us, for a healthy person, because as long as someone is healthy, the viscosity of their blood really should not change under very many conditions, unless you're talking about things like dehydration, electrolyte imbalance, having oddities in your hematocrit or your formed element numbers. So this is usually not going to be a super big deal for us, so it's worth mentioning, but in most cases we are going to assume that the blood viscosity does not really change over time. Vessel compliance is another one. So veins, we mentioned, are much more compliant than arteries, much more elastic in the idea that they can expand to accommodate a greater volume of blood. And it's a good thing that they do, because we mentioned that a majority of your total blood volume is in the veins at any given time. So the more compliant a vessel is, the better the blood flow will be and the lower the pressure. So arteries are not nearly as compliant, so naturally the pressure is going to be a little bit higher. And that's a good thing. We want the pressure in the arteries to be sufficiently high. And then another one, which is kind of fun to think about in terms of thinking about kind of differences in arterial pressure in different parts of your body, has to do with vessel length and vessel diameter. The longer and narrower a vessel is, the more resistance to flow. So the further an artery goes to service a particular part of your body, the more and more and more resistance to flow there's going to be, and the higher a pressure we need to drive blood through the entire length of that vessel. And then, of course, you are familiar with the fact that the narrower the diameter is, the worse the blood flow and the higher the pressure. So you can see a couple of helpful graphs here just to kind of get you to grasp some of these concepts. So you can see the relationship between vessel diameter and the different types of vessels that we've talked about. So arteries and veins, we've talked about, tend to have the greatest diameter, with veins having by far the greatest diameter. Capillaries have the lowest diameter. That's not surprising based on what we've talked about. So cross-sectional area, we're not really going to worry about. Average blood pressure, obviously this is something that we have extensively discussed. Arteries are going to have the highest pressure, and then as you make your way chronologically through the circulation, the pressure gets lower and lower and lower and lower, and we understand exactly why it has to be that way. And then the velocity of blood flow, meaning how quickly or how good the blood flow is in terms of getting blood circulating very quickly. Obviously, because of the high pressure situation in the arteries, blood moves pretty quickly. And because of the low pressure situation in the veins, blood moves relatively slowly. And then at the capillary level, not only is the pressure pretty low, but the diameter is pretty low as well. The blood flow is pretty abysmal at this point, so the blood velocity is going to be pretty low at that point. So a couple of the last things we want to talk about is some case study or some disease pathology type of stuff as it pertains to blood vessels itself. So atherosclerosis is something that we briefly mentioned when we talked about ischemia in chapter, I want to say, was it chapter 18 or chapter 19? I can't remember, but we have briefly mentioned atherosclerosis before. So you understood from then that atherosclerosis is basically the result of artificially narrowing the diameter of an artery because of the buildup of these fatty plaques that are the result of a number of different factors, whether it's genetics or poor diet or something else. So these plaques that you see here are actually a combination of insoluble stuff like fats and cholesterol and fatty acids and things like that. But most of what it is actually a particular type of cell, what is called a foam cell. A foam cell is actually a macrophage, which is a component of the immune system. Macrophages are good. They do what they're supposed to do. But one thing macrophages really like is they like to go around and start gobbling up lipids. So macrophages become foam cells when they have accumulated enough of these lipids that they've been grabbing from all over the place. And when macrophages do this, and when they become foam cells, they become very sticky, and they like to glom on to the sides of blood vessels, and everything can kind of precipitate from there on out. So these plaque-like protrusions obviously narrow the diameter of the blood vessels, and obviously that can produce ischemia. We can cut off the blood supply to the brain, which would produce a stroke, to the heart, that would produce a myocardial infarction, and so this is not good no matter where it happens. It just so happens the brain and the heart are going to be kind of the most severe cases of this. And another problem is that these plaques will harden the artery and lower their compliance. And you will recall when we went back to the concept of compliance here, so the more compliant a vessel is, the better of the blood flow. So if we reduce the compliance of an artery, the blood flow is going to get worse and worse. So it doesn't necessarily just have to be a diameter issue. So normally fats and cholesterol are transported through the blood by protein complexes called low-density lipoproteins and high-density lipoproteins. So something for you to think about on your own, why would we need proteins to carry around fats and cholesterol through the blood? So that's going to get back to something that we've really hammered on earlier in the semester, but think about the chemical nature of fats and steroids and things of that nature. Why would they need a carrier to take them around and chaperone them around through the blood plasma? So make sure you think about that, and we can talk about that in Collaborate if need be. So LDLs and HDLs both serve basically the same function of transporting fats and cholesterol and other sorts of things around through the blood plasma, but I'm sure you are at least aware or at least have heard of this. LDLs are the good cholesterol, I have that backwards. LDLs are the bad cholesterol because they are thought to be pro-atherosclerosis in the sense that they help to contribute to atherosclerosis, and HDLs are colloquially the good cholesterol because they are thought to go against atherosclerosis. They are thought to make things better. So LDLs are typically thought to be bad because they carry these fats and cholesterol around, and they have a tendency to get captured by the macrophages, promoting foam cell formation and contributing to the onset of atherosclerosis, whereas HDLs basically go around stealing cholesterol and fat away from foam cells, slowing down the process of atherosclerosis, plaque formation, and helping things to get better. Okay, so this is something that we talked about a little bit in chapter 19 in terms of when would you want to adjust your cardiac output for the sake of homeostasis. So let's talk about this just a little bit more now. So blood pressure, which we're going to treat blood pressure just like any other homeostatic parameter that we've talked about before, whether it's blood glucose, your body temperature, your blood osmolarity, your blood oxygen levels, your blood carbon dioxide levels, which will be things that we talk about in chapter 22, I think it is. So blood pressure is just like any other homeostatic parameter that we've talked about before. We want to keep it in a normal range. We don't want it to get too high. We don't want it to get too low. So back in chapter 19, we mentioned that the type of sensory receptor that is going to monitor our blood pressure are called baroreceptors, and these are embedded within the carotid artery and the aortic arch. So because blood flow and blood pressure are very high there, it's the perfect spot to keep an eye on and measure our blood pressure. So the interesting thing about these sensory neurons that have these baroreceptors is that they are going to produce, even when the blood pressure is normal, a baseline frequency of action potentials. So look at our mean arterial pressure as it goes from low to very high here. Our mean arterial pressure is usually going to be somewhere between 90 and 100. So let's look at this 100 here and just kind of treat it as our normal situation here. So even when the blood pressure is normal, we produce a pretty quick frequency of action potentials. But look what happens when the mean arterial pressure starts to rise, meaning that our blood pressure is getting way too high and we want to eventually do something to counter that. So when the blood pressure starts to rise, the number of action potentials gets more frequent and more frequent. So our brain, specifically the medulla oblongata, which is where that cardiovascular control center is, the way it, as the control center, is interpreting these afferent signals coming in is that when those action potentials start to get really, really, really fast, it interprets that as, okay, the blood pressure is way too high. I need to coordinate a response to bring the blood pressure down. Whereas if the blood pressure starts to drop from a normal value of 100, you'll notice the action potentials get less and less frequent. This is important because now the medulla can say, okay, these action potentials are getting less and less frequent. I need to do the opposite. I need to bring the blood pressure up. So this seems like a strange situation in which the sensory neuron is firing action potentials even when there is no problem, but it is extremely important for it to be like this because it allows for us to have a binary response range, meaning that we can respond when things get too high and we can have a different response when things get too low. So when the action potentials get more frequent, the blood pressure's too high. When the action potentials get too infrequent, the blood pressure's too low. So if this were a kind of more typical situation in which the sensory neuron is not firing any action potentials as long as things are normal, that is not going to allow the medulla to distinguish between high blood pressure and low blood pressure. Whether it's high blood pressure or low blood pressure, the action potentials would just start to ramp up. And your medulla is going to be like, okay, I know there's something wrong, I just don't know what the problem is. Having a situation in which, even under normal conditions, we have a certain baseline frequency means that the medulla can tell the difference between high blood pressure and low blood pressure. So it's very important to make sure that you understand that. So the frequency of those action potentials corresponds directly to what degree are those pressure receptors or baroreceptors being stretched. The more they're being stretched by a very high pressure, the more frequent the action potentials become. The lower the pressure, the less frequent the action potentials are. And like I said, the control center that these afferent signals are going to from the baroreceptors through those sensory neuron axons is the vasomotor control center in the medulla. So I think I showed this same picture towards the end of chapter 19. So this is going to bring us back to the autonomic nervous system and its effects on the cardiac output. We already have seen that the cardiac output is one of the most important determinants of your blood pressure, right? So what we want to do here, since we have a sympathetic response and a parasympathetic response, we want to make sure that we understand when do you want the sympathetic response and when do you want the parasympathetic response. So regardless, your appropriate response to either high or low blood pressure is going to do a couple of different things. We can do a couple of different things as we determined at the end of chapter 19. We can do a couple of different things to regulate our cardiac output. We can change the heart rate, or we can change the stroke volume, or a third option, we can change mean arterial resistance, meaning we can vasoconstrict or vasodilate blood vessels to promote better blood flow and reduce blood pressure or raise blood pressure. So what you can see here is our sensory receptors that are located in the carotid and the aorta. If there is a high blood pressure or low blood pressure situation, they will report to the medulla here, and then from there, we need to pick whether we want a parasympathetic or sympathetic response. So the basic idea here, and if some of this stuff is not making sense to you, go back to chapter 19 and review the role that the autonomic nervous system has in cardiac output, and it should start to make more sense. So the idea here is that if your blood pressure falls, you are going to want a sympathetic response. So sympathetic response is going to raise your heart rate, raise your stroke volume, and therefore raise your blood pressure. So you always want your response to be counter to whatever your stimulus or whatever your problem is. On the other hand, if you have a rise in blood pressure, you are going to want a parasympathetic response. A parasympathetic response will lower the heart rate through innervation of the SA and AV nodes, and therefore lower the cardiac output. Notice, though, that the parasympathetic response cannot change the stroke volume because there is no innervation of the ventricular myocardium, and it cannot alter vasoconstriction and vasodilation because that is a purely sympathetic response. So just as an example here, for a situation that I think all of us are familiar with, so it says here, when a person transitions from a supine or lying down position to a standing position, basically we're saying that, like, I think I've asked you all this when we were still meeting face to face. Have you ever gone from lying down in bed and you get up too fast and you feel kind of dizzy, you feel kind of woozy? So basically what's happening there is that because you went from lying down to standing up so quickly, gravity brought blood from the top of your body down into your legs because your blood pressure was at a certain level, and introducing a gravitational force allowed the blood to kind of all fall down into the bottom half of your body, into the bottom trunk. So that means less blood flow for your brain, and that's why you feel a little bit dizzy. So your body is going to interpret this as a decrease in blood pressure, an impermissible decrease in blood pressure due to decreased venous return to the right atrium, a drop in end diastolic volume, a drop in stroke volume, a drop in cardiac output. All of those things together are going to reduce your blood pressure, which again is why you feel dizzy. So what's going to happen here is that the baroreceptors of the carotid and the aorta are going to detect that drop in blood pressure. They will report that afferent information to the medulla. There's our control center. And then we hopefully settle on the fact that we want a sympathetic response here. The blood pressure is low. We want to bring it up. So, as we've said, we can do several different things here. We can increase our heart rate, we can increase our stroke volume, and we can increase total peripheral resistance through vasoconstriction and through just vasoconstriction in this case. So what you're seeing here are only two of those, although really all three of them would probably be acting together. So simultaneously, you see the sympathetic response increase your heart rate and presumably also increase your stroke volume. That increases your cardiac output. And because we know that cardiac output is directly tied into your blood pressure, that increase in cardiac output will raise your blood pressure. Additionally, the sympathetic response will vasoconstrict a lot of arterioles all over the place. That reduces blood flow and increases the amount of pressure that is needed to drive blood flow through. That increases total peripheral resistance, and that will increase blood pressure as well. Okay, so one of the last things that we're going to talk about here is capillary exchange and tissue perfusion, what are called sometimes Starling forces. So I think at this point we are aware that at the capillaries we are going to deliver nutrients, deliver oxygen, pick up waste, and other sorts of things like that. But unfortunately, when blood flows through a capillary bed like this, we said the blood flow is going to be relatively slow, but it's not that slow. In fact, the blood flow is a little bit too quick here in order for things to be delivered and taken away just by simple diffusion. So as the blood flows through, we can't just have the oxygen moving by simple diffusion. We can't have nutrients moving by simple diffusion. The blood flow is too fast for that to really be the case. So instead, what we are going to rely on here, rather than just simple diffusion, is a pressure-driven mechanism, which is called bulk flow. So basically the idea is that because of pressure differences between the blood plasma and the interstitial fluid, the pressure will drive bulk components of the blood or of the interstitial fluid to move from one place to another, either inside the capillary to out in the interstitial fluid, or the interstitial fluid into the capillary. So the direction of this blood flow is driven by what is called the net filtration pressure. So the net filtration pressure, what is called the Starling force equation, it's kind of a complicated looking thing. So there are several things to kind of digest here. You need to know 4 different types of pressures that are all measurable. The capillary hydrostatic pressure, which is basically the pressure inside the blood plasma of the capillary. The interstitial fluid colloid osmotic pressure, which is the osmotic pressure out in the interstitial fluid, that is basically drawing water into the interstitial fluid from the capillary, minus the interstitial fluid hydrostatic pressure, so the pressure of the fluid in the interstitial fluid, plus the blood colloid osmotic pressure, which is the osmotic pressure absorbing water by osmosis from the interstitial fluid. So it's a mess. This equation is a big, gigantic mess. So rather than make you try to digest this and force you to do calculations on it, although I do think there is some value in that, we can sum it up a little bit more simply. Your net filtration pressure is basically just the total pressure inside the vessel minus the total pressure outside the vessel. So whatever the number is, whether you get a positive number or a negative number, that is going to determine whether total materials are moving from the inside of the blood out or outside the blood in. And the reason why this is important is because that number is going to change as you go from the arterial side of the capillary to the venule side. So the closer you are to the arterial side of the capillary bed, that filtration is going to be coming out, meaning that bulk flow is going to cause materials from the inside of the blood vessel to move out into the interstitial fluid. So your net filtration pressure here would be a positive number driving blood flow out. So you can see here it's positive 10, meaning the pressure inside the vessel is lower than the pressure outside the pressure inside the vessel is higher than the pressure out. So bulk materials move from the high pressure area to the low pressure area. Once you get more to the venule side, as the pressure drops, you'll notice that we flip things. Now the pressure outside the vessel is higher than the pressure inside the vessel, and that's going to direct materials to move from the interstitial fluid to the inside of the vessel. So this is actually kind of interesting, and it should make some sense. We said that for a capillary bed, we are trying to accomplish 2 things. We are trying to deliver nutrients, deliver oxygen, but we're also picking up waste products that need to be taken away. So the way that I like to think about this, it's not 100% accurate physiologically speaking, but it should help you kind of think about this the right way. On the arterial side, where you are filtering things out, pushing materials into the interstitial fluid, think of that as being your vehicle for delivering oxygen and delivering nutrients. And then on the venule side, when you are absorbing materials from the interstitial fluid to the capillary, think of that as your way of picking materials up, picking up waste products like carbon dioxide and like urea. So it's really not unlike the job of a postman or a post-delivery person, right? So if you leave a letter in your mailbox that is addressed to go somewhere, the letter carrier is going to pick it up to take it away, and they're also going to drop off your mail for the day. So it's not a bad way of kind of thinking about this. And then another thing that is very interesting and worth mentioning, you would think for the sake of maintaining your blood volume, you would hope that the amount of fluid coming out on the arterial side would be approximately equal to the amount coming in on the venous side. If we keep those things perfectly balanced, we should not gain or lose any material into the blood. Well, as it turns out, we actually lose more fluid out of the blood on the arterial side than we get back on the venous side. So there is a discrepancy here. If there is nothing to account for this, every time blood moves through a capillary, we lose some of our blood volume into the interstitial fluid. So we need something to make up that difference there. So the answer to that conundrum is that the lymphatic system, which is something we haven't talked about a whole lot, can pick up that extra fluid accumulation in the interstitial fluid and then eventually return it to the veins before we get back to the right atrium through the vena cava. And a perfect clinical example of how and why things like this can go wrong is what you know to be swelling, also called edema. So the kind of drastically severe case that you're looking at here is a disease called elephantiasis. Elephantiasis is caused by parasites which are carried by a mosquito vector. These parasites can clog up the lymphatic drainage that we just cited, meaning that because the lymphatic system can't return that excess fluid to the veins, that fluid instead will accumulate in the interstitial fluid of the tissue, resulting in this severe edema. So all the swelling that you are seeing right here is just a drastic increase in the volume of the interstitial fluid as it accepts fluid from the blood that flows through on the arterial side, and we never are able to return that excess fluid into the veins. So in addition to that, edema can be caused by any of the following, and all of these are going to play into the Starling forces that we just got done talking about. So if we mess around with that equation in any particular way that either promotes extra fluid loss or inhibits fluid gain on the venous side, you can end up with edema, this fluid accumulation in the tissue. So number one, high arterial blood pressure will do this. So this is going to raise the blood colloid, the capillary colloid pressure. So that is going to cause increased filtration of fluid out on the arterial side, and we have no mechanism for making that up on the venous side. So people with high blood pressure tend to get a little bit of swelling in some of their extremities. A venous obstruction will do this, so this will raise the capillary hydrostatic pressure, meaning we get more filtration out again. An obstruction of the lymphatic drainage, we saw that as an example with elephantiasis. And then number 4 here is kind of the biggest one. So leakage of plasma proteins into the interstitial fluid. So raises interstitial colloid pressure at the expense of plasma colloid pressure. So that's a lot of mumbo jumbo, right? That's a lot of gobbledygook. Basically, number 4, that's injury. That's tissue injury. So if you have some sort of injury, whether you sprain your ankle, you do something to injure yourself, what you are doing with that injury is you are destroying some cells, and those cells are going to leak all of their proteins out into the extracellular fluid. That raises the colloid pressure of the interstitial fluid, and it's going to start changing the fluid dynamics such that any capillaries that are servicing the injured area are going to start leaking their fluid in there, and that fluid is all going to start accumulating there. Okay, so that does it for Unit 4. We are all done at this point. Congratulations. All you have to do now is take the exam that goes along with this. So good luck with that. You still have a week or more to study for that. So I will go ahead and sign off for now, and I will see you later.